In this section, we're going to talk more about endocrine signaling and the different ways that it's done. We're going to start with talking about the specific receptors that receive signals. So there are internal receptors that are actually often located inside the cytoplasm of the cell. And these bind to ligands that are hydrophobic. Remember, hydrophobic doesn't like water. And you remember that the inside of the plasma membrane, the fatty acid tails of the phospholipid bilayer, that part is hydrophobic. So a hydrophobic signaling molecule or ligand can go straight through the plasma membrane. It's like, yeah, hey, you're a fat, I'm a fat too, let me by. And then once it gets inside the cell, it binds to an intracellular receptor. Intra means within. So this is a receptor within the cell. For those of you who are wondering, well, how does it know which cells to go into? It doesn't. It just goes into cells, and some of those cells have the right receptor. In some others, it wanders around wishing that there was someone there and not finding a friend, and that's sad for it. In this case, the receptor and the ligand together stay bonded together. And this is often a signaling pathway that can turn genes on and off. And so it will then go into the nucleus, bind to DNA, and copy or transcribe DNA so that a protein can be made from it. So in addition to lipid hormones, there are some gases that operate this way, including nitrous oxide. Nitrous oxide is, of course, laughing gas. It is a pain reliever that I guess dentists used to use. They don't use it very often anymore. But it is also released by some cells in your body, and it can go straight through the plasma membrane. So those are internal receptors. Most commonly, though, receptors are cell surface receptors, meaning that it's a protein that's embedded in the cell membrane. So here's some examples of some different proteins embedded in the cell membrane. And you can see we've got a receptor here that has a binding site for a ligand. Now some of these are going to open an ion channel like this one, like the paracrine receptors. Okay, even though that's not endocrine signaling, it still uses a receptor. So for example, in a neuron, the neurotransmitter would bind to the ion channel, the channel opens, and a different molecule or a different ion goes through. They can also be transporters, or they can start a transduction pathway, which is what we're going to talk about some more in a minute. Transduction just means a continuation. All right. Cell surface receptors have 3 different parts, which are super obvious, but for some reason someone decided to call them domains. So we have an extracellular domain, which is outside of the cell, a transmembrane domain, which goes across the membrane, and an intracellular domain, which is inside the cell. Like, duh, what else would it be? Some of these are also connected to other proteins that are going to be involved in the cell response. And in that case, that other protein is going to be in the intracellular space in the cytoplasm. There are 2 main classes, of the 2 main classes of receptors. There are a few kinds of receptors, ion channel linked receptors are going to open an ion channel. So this is where the membrane protein binds to some signaling molecule, which is the ligand, and then opens and allows ions to either flow into or out of the cell. These are really, really important, both in the function of your muscles and your neurons. So those are important. Yeah. None of this requires the use of ATP. Okay, these are going to bind. The binding is going to cause a conformational change in the protein, which opens the channel. And the ions are going to move down a concentration gradient. Now, a big group of receptors are called GProtein linked receptors or GProtein connected receptors. These are called that because they are linked to a G protein. Yeah, okay. And then this is going to start some more reactions, which is what we call a signal cascade. All G protein linked receptors, for some reason, have 7 transmembrane parts. Why? It's just the G protein linked receptor thing. They all go across the membrane 7 times. They have an extracellular portion, they have an intracellular portion, and they have 7 transmembrane domains. All right, so here is a diagram showing a G protein coupled or linked receptor bound to the heterotrimeric G protein. So this whole thing is the G protein, okay? But it's actually 4 parts. It's actually 3 different proteins plus a GDP, not the gross domestic product. It's like ADP, but with a guanine instead of an adenosine. Okay, remember we talked very, very briefly during cell respiration about GTP? Here it is. It still gets made in the mitochondria. Okay, so this sits in the cell membrane. These 3 proteins and the GDP bind together, bind to the receptor, and wait for a ligand. The ligand, or the signaling molecule, binds to the receptor. And when that happens, the protein changes shape, and that opens up a site where a GTP can bind. The GTP binds, and the GDP is released. This is weird to me that it doesn't just phosphorylate the GTP or the GDP, but whatever. That's not what it does. Then the 2 of the parts break off from the first. So the first is called the alpha unit. The other 2 are the beta and gamma units. The alpha unit and beta gamma units let go. And then the beta and gamma units go into the cytoplasm and bind to other things and start another series of reactions, all of which cause a cellular response. Now, there are some toxins that inhibit the GCPR or bind to the GCPR and prevent the regular ligand from binding to it so that the normal signals don't get sent. The toxin that causes pertussis, botulism, cholera, all of those interact with G protein linked receptors. Botox is a very, very dilute form of the botulinum toxin created by a bacterium. And so in Botox, what you're doing is you're injecting a very, very dilute amount of toxin into muscle cells, inhibiting the receptors on the muscle cells that receive the signal to contract and paralyzing the muscle cells. So that's why Botox gets rid of wrinkles, because it just relaxes the muscles whose muscle tone or sort of constant small level of contraction causes wrinkles in your face. But that's also why people who have had Botox, like if they've had Botox on their forehead, they can't look surprised because they can't raise their forehead because the muscles have been paralyzed. Yeah. It's also why nurses have to be trained really, really well, because if you don't dilute the Botox enough, you can inject someone with too much botulinum and kill them. So yeah, be careful out there. All right. Finally, the GTP does get hydrolyzed, releases a phosphate group. That changes the shape of this alpha unit. The alpha unit goes back, reconnects with the G protein receptor, and then beta and gamma come back and rebind to alpha, and the whole thing is back into its original shape and can do the whole thing again. Okay, so this is a really long explanation of G protein linked receptors. They are really important though, and they are present on just so many cells. Okay, so I'm going to stop this here, and then in the next section, we're going to talk about some other receptors.